Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with polyol pathway of glucose. In today's session, we will start with the fructose metabolism, in which we will compare it with glucose metabolism. Then in fructose metabolism, we have azoospermia, its role in myocardial infarction, hereditary fructose intolerance, and fructosuria. Then we have galactose metabolism, in which we will study that UDP glucose and UDP galactose are interconvertible. Lastly, we have galactosemia. Good morning, everyone. Let us start the topic. Fructose metabolism. Fructose is a ketohexose present in fruits, honey, and sugar, that is sucrose. Soft drinks have the sweetener corn sugar, which has a high fructose content and is sweeter than sucrose. Fructose is promptly metabolized by the liver. Fructose is phosphorylated by fructokinase, an enzyme present in birth with a high affinity for fructose. In this diagram, we can see fructose metabolism. Fructose is converted to fructose 1 phosphate with the help of enzyme fructokinase, which is absent in fructose. Then this fructose 1 phosphate is converted into glyceraldehyde with the help of enzyme aldolase B, which is absent in hereditary fructose intolerance. Then this glyceraldehyde plus dihydroxyacetone phosphate together they form glyceraldehyde 3 phosphate with the help of enzyme triose kinase with the conversion of ATP to ADP. Fructokinase phosphorylates the substrate at the first position, whereas hexokinase action is on the sixth position. Fructose is not dependent on insulin, so fructose is more rapidly utilized in the normal persons because glucokinase and phosphofructokinase, that's so-called metabolic bottlenecks, are not encountered in fructose metabolism. Therefore, in theory, fructose will be better utilized in patients with diabetes mellitus because the first few enzymes of fructose utilization do not require insulin. But in experiments, fructose was found to be deleterious in patients with diabetes mellitus. This is because fructose rapidly enters the tissues and leading to increased fatty acid synthesis, increased serum triglycerides, and increased LDL cholesterol level in the blood. And all these are atherogenic and harmful in nature. Fructose metabolism is in liver bypasses the PFK control point. Hence, fructose increases the flux of glycolytic pathway leading to lipogenesis. Moreover, glycerol phosphate required for triacylglycerol synthesis is provided by the metabolism of fructose, leading to the increase in TAG pool in the body. Phosphorylation of fructose by fructokinase depends on the cell of ATP. In the low ATP level, increases oxidative phosphorylation leading to lowering of Pi levels in the cell. This will remove the diminishing effect on adenosine deaminase, and there is increase in the rate of conversion of AMP to IMP and then to uric acid. Accumulation of fructose 1 phosphate also inhibits glucogenesis. In muscle, fructose is phosphorylated by hexokinase to fructose 6 phosphate. The fructose 1 phosphate is then cleaved by the enzyme fructose phosphate aldolase or aldolase B. The products are glyceraldehyde and dihydroxyacetone phosphate. Fructose is mainly metabolized by liver but free fructose is seen in large quantities in seminal plasma. The energy for mobility of spermatozoa is mainly derived from fructose. Fructose is secreted by seminal vesicles. In some persons, azoospermia is seen due to a block in the duct. In such persons, fructose is estimated in semen. In this diagram, we can see that seminal vesicle is a place where fructose is secreted. High fructose levels are known to be related to aging and myocardial infarction. Fructose participates in glycation 10 times faster than glucose, and the decreased antioxidant contribute effects to ischemic heart disease. In this diagram, increased fructose metabolism leads to blockage in the blood and finally leading to ischemic heart disease. Coming to the uh, hereditary fructose intolerance, it is an autosomal recessive inborn error of metabolism. Incidence of disease is 1 in 20,000 births, while 1 in 70 persons are carriers of the abnormal gene. The defect is in aldolase B, hence fructose 1 phosphate cannot be metabolized. This is seen when sucrose containing fructose is introduced in the diet of infants usually around 6 months of age. Accumulation of fructose 1 phosphate will be glycogen phosphorate, inhibited by glycogen phosphorylases. This leads to accumulation of glycogen in liver and associated hypoglycemia. Vomiting and loss of appetite are seen. The infants often fail to thrive. Hepatomegaly and jaundice may occur. If liver damage progresses, death will occur. Fructose is also excreted in urine, which gives positive Benedict's test. Withdrawal of fructose from the diet will immediately relieve the symptoms. This is coming to the next topic, fructosuria. This is a benign metabolic defect due to the deficiency of fructokinase. There is no abnormality other than excretion of the fructose in urine. Fructose is a non-essential, not a dietary essential. Urine gives positive Benedict's as well as Selivanov's test. Incidence is 1 in 1.3 lakh births. Coming to the next cycle, galactose metabolism. The term galactose is derived from Greek word gala, which means milk. Galactose is an aldohexose and is the fourth epimer of glucose. Galactose is a constituent of lactose of milk sugar and is taken in the diet. Galactose is not an essential nutrient because UDP glucose can form UDP galactose. In this diagram, we can see galactose metabolism. Galactose is converted into galactose 1 phosphate with the help of enzyme galactose kinase and conversion of ATP to ADP. This galactose 1 phosphate is then converted into UDP galactose with the help of enzyme galactose 3 phosphate uridyl transferase and conversion of UDP glucose to glucose 1 phosphate. In those with galactosemia, this this reaction will be absent. Then UDP galactose is reversibly converted into UDP glucose with the help of enzyme UDP gal epimerase. There is one more pathway for UDP galactose, which is by the enzyme which is 4 to 5 years occurs in the life. Galactose 1 phosphate require is converted into glucose UDP galactose and conversion of UTP to PPI along with the help of enzyme galactose 1-phosphate biophosphorylase. Galactose is metabolized almost exclusively by the liver, and therefore galactose tolerance test is done to assess the functional capacity of the liver. UDP galactose is the active donor of galactose during synthetic reactions. Galactose is necessary for synthesis of the following. First, it is utilized in lactose synthesis. UDP glucose converts into UDP with the help of enzyme galactose epimerase. Then UDP galactose plus glucose give lactose, which is due to enzyme galactose synthesis. Lactose synthesis is seen in lactating mammary glands. It is also useful in synthesis of glycosaminoglycans, cerebrosides, lipids, glycolipids, and glycoproteins. The step 1 of glycolysis, galactose metabolism. Galactokinase reaction. Galactose is first phosphorylated to galactokinase to galactose 1 phosphate. The next step is galactose 1 phosphate uridyl transferase. This is the rate limiting enzyme in the galactose metabolism. UDP galactose may be used as such for synthesis of compounds containing galactose. Next step is epimerase reaction. By this reaction, the galactose is channeled to metabolism of glucose. Since the reaction is freely reversible, even if the dietary supply of galactose is deficient, UDP glucose can be epimerized to UDP galactose. There is alternative pathway. The galactose 1 phosphate pyrophosphorylase is in the liver becomes active only after 4 or 5 years of life. The enzyme will produce UDP galactose directly, which can be epimerized to UDP glucose. Coming to galactosemia, there is deficiency of enzyme galactose 1 phosphate uridyl transferase. It is an inborn error of metabolism. The incidence is 1 in 35,000 births. Due to the block in the enzyme, galactose 1 phosphate will accumulate in the liver. This will diminish galactose kinase as well as glycogen phosphorylase. Hypoglycemia is the result. Bilirubin uptake is less and bilirubin conjugation is reduced. So unconjugated bilirubin level is increased in the blood. There is enlargement of liver, jaundice, and severe mental retardation. Free galactose accumulates, leading to galactosemia. It is partly excreted in urine. Galactose is reduced to dulcitol. The accumulation of dulcitol in the lens results in cataract due to its osmotic effect. It is called congenital cataract and is very characteristic feature of galactosemia. Galactose 1 phosphate may be deposited in the renal tubules, producing tubular damage leading to generalized amino aciduria. Coming to the diagnosis of galactosemia, clinical manifestation including congenital cataract and presence of galactose in urine as well as in raised blood galactose level will help in the diagnosis. Collection of fetal cells by amniocentesis may be useful in perinatal diagnosis. Heterozygous patients could be detected by elevated galactose levels in blood after galactose load. Coming to the treatment of galactosemia, if lactose is withdrawn from the diet, most of the symptoms recede, but mental retardation, when established, will not improve. Hence, early detection is most important. For infants affected, lactose-free diet is given. Such patients may be withdrawn after 4 years. when galactose 1 phosphate pyrophosphorylase becomes active. Here ends our channel. Kindly like, share and subscribe the channel. Thank you.